Dear students, welcome to the lecture number 7 of Digital Image Processing. Today's topic is biological aspects of image acquisition. So after the completion of today's lecture, you will be able to understand about human visual system and the properties of human visual system. In the previous 6 top lectures, you have studied about the fundamentals of digital image processing, nature of digital image processing, its applications, digital image representation, types of images, file formats. So all these topics you have studied. So let us start today's lecture. Biological aspects of image acquisition. You know that most of image processing applications produce images for human observation. So it is very important to understand the characteristics and limitations of human visual system. Human visual system consists of mainly 3 parts: eye, optic nerve and brain. Human eye can be easily compared with the camera system. So in terms of camera system, eye can be related with the image sensor or camera, optic nerve with the transmission path, and brain is related with the image information processing unit or computer. Here you can see the structure of the eye. Human eye is the most complex organ of human body. It is spherical in shape and its diameter is approximately 20 mm. The human eye can be compared with the camera system. Like a camera, it converts visual information into the neural signals which is processed to the brain, and accordingly image formation can be done and we can visualize the image. So our eyes enables us to see all the beautiful things around us. In this lecture, you are going to study about the structure of human eye, its components and their functions. You can see the components. This is iris, this brown part, and on this brown part there is a black portion which is known as a pupil, sclera, optic nerve, cornea. You have studied the structure of human eye in your school as well, but it is very much important to understand and to recall all the components and its function to understand the image processing applications. What is the layer of the eye? There are 3 layers in the eyes known as cornea, which is the outer tough transparent membrane. Another layer is sclera, which is again the outer tough, but this is opaque membrane. And the third is choroid, which contains blood vessels and provide nutrition to the human eye. So these are the 3 layers of the eye: cornea, sclera and choroid. Here you can see a cross-sectional anatomy of the human eye. So it will help you to understand the function of each and every part. If you have to concentrate only on the eye and discard all the surrounding skin, all the things, then you can see a ball which is known as an eyeball. In eye, the most visible part of eye is iris. Sometimes you talk about the color of eye, which is actually the color of iris. So when light hits our eye, it falls onto our iris and gets reflected. So it totally depends upon the pigment that makes it up. If it is green in color, then you can say that the person has green eyes. If it is brown in color, you can say the person's eyes is brown in nature. If you notice iris, you can see a hole into center, and this hole is known as pupil. Through the pupil, light enters into the eyes, and this is what enables us to see the things. Our iris has a very important job. Its job is to control the size of pupil. Iris is actually controlling the size of the pupil. Our iris will make hole smaller during the daytime when the ambient lighting conditions are very bright. On the other hand, during the night or when you are inside into a dark room, so you would like to open up pupil, otherwise it would be difficult to see anything. So actually this iris is only which is controlling the size of the pupil. Here you can see a bulgy part of the eye which is in front, and it is known as cornea. And over here you can see a lens which is known as a lens of the eye. You can see the lens. The space between the cornea and the lens is filled with a watery kind of liquid which is known as aqueous humor. This is watery kind of liquid. And the back part of the eye is completely covered with light sensitive cells. It is known as retina. Light sensitive cells convert light into the electricity, and finally these electrical signals are carried from retina to the brain through some nerves. From retina to the brain. These nerves will connect all the cells of retina, and this bundle of nerves are known as optic nerve. Here you can see so many nerves are there, and this whole bundle is known as the optic nerve. Our brain is super complicated, but is very interesting if you try to understand it. There is a space between the lens and the retina which is known as vitreous humor. It is filled with a jelly kind of substance. Means it looks like a glass, but actually it is not glassy in nature. It is actually made up with organic substance. You know that the retina is a eye sensor system, and the distance between the lens and the retina varies from 14 to 17 mm. The description about iris, pupil, lens and retinas I have told you in detail. Here you can see a diagram which shows how the amount of light is being regulated by the human eye. Various components of human eye have also been shown over here, like iris, pupil, cornea, sclera, optic nerve, retina. So iris actually open and closes to control the amount of light entering into the eye. If you compare it with a camera, it is like a diaphragm in microscope. Pupil is the hole of iris where light enters into the eye, and in camera it is like a aperture. The iris acts as a variable aperture to control the amount of light that is allowed to pass through the lens, and this is actually controlled by ciliary muscles which open and close, therefore controlling the pupil for focusing. In general, you can say that the curvature and degree of refraction of the lens are adjusted so that we get a sharp image. Here you can also see a retina. It is a eye sensor system. You know that the distance between the lens and retina is approximately 14 to 17 mm. Retina is composed of photoreceptors. There are two basic types of photoreceptors, cones and rods. Image formation by the eye. A very simple process. You can see over here, a cross sectional of eye is being shown over here, where eye collects the light and focuses on the retina. Therefore, image formation is being done. Retinal photoreceptors. If you talk about the photoreceptors, you know that there are two types of photoreceptors known as cones and rods. So when light strikes on the cones or rods, it creates electromechanical reaction that generate the neural impulses. In this diagram, you can see how cones and rods are connected to a nerves. See, if this is the black portion of the eye, and here you can see that cones and rods are connected to the human nerves. Rods are long in structure, like here rod is being shown, it is long in structure. Rods are more sensitive to the light, and approximately 6 to 7 millions of rods is located in the central portion of the retina. If you talk about cones, they are generally shorter and thicker in shape. They are less sensitive to light, and approximately 75 to 150 millions of the cones are distributed over the entire retina. Cones are useful in bright light vision, while rods are useful in low light vision. You know that human eye has 3 different types of cones, and each cone responds to a distinct spectral band of light. So it creates the perception of colors, because cone respond to the generating a human set of response for each color of light. So these responses form 3 types of cone, and brain forms a distinct perception of color. And you know that this distinct perception of color is known as a concept of trichromacy. How rods and cones operate differently? From this particular diagram, you can see that the distribution of rods and cones are not evenly around the retina. It varies in numbers, works on the intensity levels as well. Cones have different absorption characteristics as a function of light. So accordingly, rods and cones are connected to the nerves. Rods responds to low level of illumination, which is known as scotopic vision, while cones respond to the high level of illumination, and this is known as the photopic vision. Next topic is properties of human visual system. You know that light is a physical phenomena. It can be measured. Light is dependent on the surface and the angle from which light is emitted. Brightness is a psychovisual concept. Its concept is the perception or sensation of the input light in the brain that produces the concept of brightness. You have also studied some other terms like contrast, which is defined as the difference of the perceived brightness. You know the term of simultaneous contrast, and here you are going to study about the Mach bands as well. So let us proceed with brightness adaptation. What is brightness? What do you understand by brightness? Brightness means intensity as perceived by human visual system, and brightness adaptation means human visual system can operate only from scotopic to glare limit. There are 2 terms, one is scotopic and one is glare. So the least amount of light that can be perceived by human eye is known as scotopic limit. And the maximum amount of light that can be perceived is known as the glare limit. Human eye can adapt to an enormous range of light intensity level, almost of the order of 10th of magnitude. Brightness perceived is subjective in nature, and in fact its variation is logarithmic in nature as a function of light intensity. This is due to the fact that brightness depends not only on the luminescence of object but also on the surrounding. Human eye cannot simultaneously operate over such a range of intensity levels. This is accomplished by changing the overall sensitivity known as brightness adaptation. Let us understand from this particular diagram. It shows the variation of log of intensity to the subjective brightness. Here you can see that the intensity level, here the subjective brightness. If we start from left to right, here the intensity level is low, as well as we are moving towards the right, intensity level increases. You can see that at this particular point, if you talk about intensity level is very low, while at this particular point intensity level is very high. In general, if you take an example of if a human or a person enter into a dark room, then the type of perception will be little bit different. If immediately that person moves into the lighter room, the perception will be the different. So a person cannot visualize both the things simultaneously, the darker one and the lighter one. That is why we are saying that it is not able to cover the entire range. Means from this particular to this particular point, somewhere adaptation needs to be managed. If we talk about this particular point, human visual system may be able to visualize the area nearby this particular point, but not from here to here. So range of subjective brightness that I can perceive when there is an adaptation in a particular level. Next is brightness discrimination. Brightness discrimination is the ability of eye to discriminate between changes in the brightness level. The increment of intensity of delta IC that is discriminable over a background intensity of eye is measured. How it is being done? Let us take an image. This is the image eye. Out of this image, let us take a small section. We say that a small circle is being taken. If we try to minimize or maximize the intensity of this particular circle while keeping the background same, there must be a variation. If we are increasing the intensity, let us say it is being increased to I plus delta I. So that was incremental is being done, and this ratio of delta I over I is known as a Weber ratio. And because of this, when you are increasing this particular intensity level, it will be able to extract and visualize properly out of this whole background. This is the concept of brightness discrimination. Smaller the value of Weber ratio, which is the ratio of delta I upon I, gives good brightness discrimination, and small change in intensity, while large value of Weber ratio gives poor brightness discrimination. So it is always being required to keep a threshold to maintain properly whenever there is a requirement to change the brightness level among the picture itself. At higher intensities, brightness discrimination is good because Weber ratio is small in comparison to the low intensity. Next term is simultaneous contrast. To understand this particular term, let us take an example of white object. If you take a white object and perceive it on a black background, and similarly taking a white object and perceive it on a brown background. Though in both the cases the luminescence of object is same, but perception will be different. One is on black background and other is on the brown background. So because of this, a result is being created, and this phenomena is known as a simultaneous contrast. So simultaneous contrast is a visual perception phenomena. A region's perceived brightness is not only a function of intensity, but it depends on the background intensity as well. In this particular image, you can see that there are three squares. Its background is black. It is having gray background, lighter background, but out of these three you can see that this particular square, smaller one, is more visible. So means you can say that all the centers of these squares having the exactly same intensity, but they appear to the human eye to become darker because of the background variation, and it is having a brighter variation, lighter intensity component, so looks good. So this is the concept of simultaneous contrast. What is Mach band? Another very important phenomena. Mach band is actually based on the perceived brightness, and it is not only a simple function of the intensity. Mach band effect is phenomena of lateral inhibition of rods and cones, where the sharp intensity changes are attenuated by the visual system. You can understand from this diagram. Here you can see if you move from left to right, there is a variation from black to white. In between there are various gray shades. So if you represent its level, black will be represented by like this, then gray like this, again lighter gray, its variation will be like this, more lighter gray, more the amplitude, and this white one will be having a higher amplitude. So if you draw, you will be getting this kind of staircase image, but you will not be able to visualize in the same manner. You will be able to visualize in this manner where you are finding the spikes. So what is the fundamental behind this? Let us take these two patterns, these two stripes. More darker, less darker. So if you observe at this particular edge, there is a black portion, darker one, intensity level is being represented by lower level, this one, while the intensity level of this is being represented by this level. But how this variation is being represented? You can see, as you see from this gray portion towards the black, you will see that this is more darker, but from when you see from black to the gray, you will find that this is more lighter. That is why you can observe a peak over here. Similarly, if you can see from here, one level is this, another level is this level. Like if you talk about these two stripes. Again, observing from here to this end, it will looks more grayish. If you observe from here to here, it will looks more bright. So accordingly, these variations when you are observing from left to right or right to left means observing from darker region towards the whiter or whiter region towards the darker. So these kinds of patterns actually being formed. So this is actually the concept of Mach band effect, which is being visualized while processing digital images. Mach band effect is used to measure the impulse response of the human visual system in spatial coordinates. In spatial coordinates means if you are writing a 2D image like fx, y, then x and y are the spatial coordinates. Next, we'll be talking about the monochrome and color image models. A model for human visual system based on its properties can be constructed. Since there are number of factors which defines the quality of photoreceptors, quality of eye optics, transmission and processing of visual information, and therefore they finally impact the quality of the image. This diagram shows the mathematical model for monochrome human visual system. Here you can see that the effect of light on the eye system using mathematical expressions, how it is being written, I of xy lambda f of xy. This model shows that the light entering into the eye first encounters with the eye optics. This block is nothing but a low pass filter, and its frequency response represents the optical characteristics of the human eye. The spectral response is the representation of relative luminous function, and the output of this block is, which is the input for the another function which is having a luminous distribution. So you can see that the non-linear response of rods and cones for the function fxy is the contrast, and the lateral inhibition is a linear system whose output is a neural system signal which is taken into the human brain through optics nerve. This model is known as a monochrome vision model. This monochrome vision model can be extended to construct the color vision model as well. A ray of light has three components, R, G and B. These RGB components are converted to retinal cone stimulus coordinates. This is required to remove the redundancy which is present in the signal. In any computer, it can be achieved using the matrices. Actually, the human eye and brain together translate light into the color. Light receptors within the eye, which are cones and rods, transmit messages to the brain, and which produces the familiar sensation of the colors. This is the mathematical model for the color human visual system. In this diagram, you can see that non-linear transforms are applied to the input. Again, light is being imparted on the object, then it is being processed, and again non-linear transformation is being done. There are two matrices A and B. Logarithmic of these like apply are the output of the H1, H2, H3. Here you can see that the components of H1 is the monochrome part that represents the intensity component, and the components of H2 and H3 are chromatic components. These are chromatic components. These are specially filtered and then sent to the human brain as inputs for color image representation. So, in the case of color model, in this way image is being processed so that human brain will be able to receive the inputs, and accordingly image can be reconstructed, which will be visualized by the human visual system. So this is about the monochrome and color image model. So students, in this lecture you have studied about the human visual system, and properties of human visual system in detail. Thank you, students.